So the other day we went over the 3 points of the cell theory. Okay? And those 3 points were that all organisms, all living organisms, are made up of cells. Okay? Cells carry out the basic functions of the organism, and cells come from the division of existing cells. And then we talked about the 4 important people really, really quickly. We talked about Hooke, okay, who was the physicist who built the first microscope, okay, and was the first person to view cells. Obviously, the cells he looked at were no longer living, so they looked empty. Hence the name cells, because they look like rooms in a prison. Okay? From there, the design of the microscope changed. Luckily, this design did not catch on because it was very hard to use, but it was also very effective. Okay? And Leeuwenhoek was able to look at cells that were alive and found that they were not, in fact, empty little rooms, that they were very full and very busy little places. And then Schleiden and Schwann came along. Okay? These were 2 Swedish scientists who worked together. Okay? And they worked on the hypothesis that if cells are the basic units of life and tissue from various types of organisms is studied under a microscope, then cells will be found in all of the samples. Okay? So simple if and then hypothesis, just like we would have had. And that's what they found. Okay? Years of study looking at a wide variety of different types of organisms. Okay? From, animals to plants to fungus to bacteria, single-celled organisms, all kinds of different things. Okay? And they found that, in fact, every living organism they studied was, in fact, made of cells. Okay? Either unicellular or multicellular, they were all, without exception, made of cells. Now, of course, at that time, nobody knew about viruses yet. Viruses were not discovered until the electron microscope was invented, because they're just too small for us to see. So they had good equipment. Magnifies up to 600 times, okay? Which is like even better than what ours do. Okay? But they still obviously weren't able to see viruses. But they were able to see cells in all of the tissue and organisms that they studied. All right, so obviously technology has improved since then, and our microscopes have gotten better and better to the point now where we can observe the processes that go on in individual parts of a cell. Okay? The little organelles like the mitochondria and the nucleus and the lysosomes and the Golgi apparatus. We can actually observe those things, okay, and see what they do, which has allowed us to understand how a cell operates, okay, how a cell kind of reproduces, how a cell does basically everything that it does. We are actually able to observe it doing it. What we've also noticed is that all cells are very, very similar. And this is something that even Schleiden and Schwann noticed. Okay? Not only did all organisms have cells, but all organisms had very similar cells. Okay? Like, we look at a plant and we look at an animal and we're like, they're very, very different. Okay? And when we look at their cells, there are some obvious differences. But there's way more the same than there is different. Okay? If we put a plant cell and an animal cell side by side, I mean, yes, the differences are the things that kind of catch our eye, but it's the similarities where, well, there's a lot more of them. Okay? There's a lot more similarities than there are differences. Okay? Both cells have a nucleus. Okay? Both cells have rough endoplasmic reticulum. Both cells have smooth endoplasmic reticulum. Both cells have a Golgi apparatus. Both cells have mitochondria. Both cells have cytoplasm. Both cells have like all kinds of stuff that's the same. Really, there's only a couple things that are different. Animal cells have centrioles. Plants don't. Plants have chloroplasts in the cell wall. Animals don't. Okay? And that has to do with where they get their energy from. Plants are obviously photosynthetic and don't move. So they can have stiff cells to support their shape, okay? And they need chloroplasts to carry out photosynthesis. animals move about to capture their food. Okay? So they need to have cells that are flexible, so that they can change shape as they move. Okay? They don't need chloroplasts, okay? Because they don't carry out photosynthesis. All good? Yep. So they're going to have some structural differences. Okay, so, but there's a lot of similarities, and that's kind of the point we're trying to get at here, okay, is that there's more similarities than there are differences. So what does that mean? Okay, if we look at a fungus, it's the same thing. Their cells have a few differences, but in general, they're still pretty much the same. If we look at a single-celled organism, like an amoeba or a paramecium, cells are still pretty much the same. So, which is a more likely scenario? They all evolved to have cells separately that were alike, or they had a common ancestor who had cells that looked like that and they diverged from there. Which one's more likely? Divergence, definitely. Okay, it's not by accident that if you look at pictures of your parents when they were your age, pictures of your grandparents when they were your age, and so on. Okay, that there's similarities. You come from the same place. You share a lot of genetic material with those people. You're related to them, right? Well, the same is true for plants and animals. They're related, be it very distantly. Okay, this would be like their 400th cousin, 100 times removed, but they're still related. Okay, that's why they have all of this similar structures and all these similar processes. We don't even see the processes. We're just looking at the structures right now. Okay, but all the stuff the cell does to stay alive, okay, a lot of that is similar too. How plants and animals make protein is the same. All right, so it's important that we understand that yes, plants and animals have evolved from a common ancestor in the very distant past. And the fossil record has even allowed us to essentially trace it back, okay, to some of the first organisms that were alive. Now, I have a lesson later on in the unit where I'll go over why evolution is not like counter to what the church says and what you've learned in religion class, why they actually do get along. Okay, but that's not for today. Okay, it'll be for another day. Questions on the similarities? Everyone's good with that? Okay. So we will be going over the parts of the cell next week, because obviously that's going to be a big part of what we do in this unit is knowing what all the parts do. Okay. All right. Now for microscopes, I'm going to give you a 2-minute break because I forgot to go grab a microscope. So I've got to just go grab one so I can explain this stuff to you here in just a second. So the microscope. Okay, when you go into the lab on Monday, we're doing our first lab with microscopes, okay, you're going to find the microscopes either have a plastic cover that's the one they came with, or our budget replacement, large Ziploc freezer bags. Okay, they also work great to keep dust off. Okay. So, and they cost a lot less. Then you'll also see a power cord, hopefully wrapped carefully around the microscope. Okay. You'll probably notice something about all of those cords. They're all messed up. Okay. Most of them have different plugs on the end. Okay. Reason for that is people don't carry them right. People don't handle them safely. Okay. The reason we wrap this thing around there is so that when I'm carrying the microscope, the cord is not dragging along on the ground like it is right now. Okay? Because eventually what happens is it gets under my foot, and then I walk this way, and the cord tightens, and one of two things happens. Hopefully the cord gives away. Okay? Because if it does, that means you're still holding the microscope. Okay? Or it pulls right out of your hand and the microscope goes crashing to the ground. The crashing sound will be followed by the ambulance sound. Don't drop these. Okay? They're really expensive and they're really delicate. Okay, so make sure you carry them properly. That carrying properly is the way you just saw me carrying it. Okay, with either the cord wrapped completely around, or in my hand, one hand on the base, one hand on the arm. This is a two-handed carrying object. Okay, it is not something you carry in one hand and swing wildly as you go.
Is the sound of the ambulance for the microscope or for the person that dropped?
I'll let you come to that conclusion. Which one would need one? Okay? All right, and of course I'm kidding. But yeah, we want to make sure we're careful with these, okay, because they are quite delicate. And they're probably quite a bit different / way better than the ones you had in junior high, okay? Because if you went to John Paul II, we know what's there. It's the ones we left there in 2005 when we moved up here. So they were junk, and they probably still are. These ones, okay, are much better. Not that they magnify a lot more, because they actually have the same lenses, but they're better in that the stage, which is where you put the slide, is actually mobile. So you don't have to put your fingers on the stage and move the slide around with your fingers. Okay? There's a couple of reasons why that's bad. Okay? First thing is, it's almost impossible to remove your thumbs from the slide without the slide moving as you move the thumbs and then you losing the focus. Okay? And the other part of it is, usually when you're moving it around, you push the stage and the stage moves and it goes out of focus. Okay? So this way, with these little knobs, okay, if I turn them, it'll move the stage and the stage clips, so that I don't lose the focus, okay? And that's really nice if I've got it in focus and I want to check out either other parts of the slide, or if I'm on high magnification, other parts of the cell, okay? Things like that. So the stage is movable, okay, with these little stage control knobs right here, right? And there's stage clips for the slide. So you pull this to the side, put the slide in here, let it go, and it'll hold the slide in place, and then it moves around when you move the stage. Okay, very good with that. All right, the other knobs, these two, these are the focusing knobs, right? This is the coarse focus knob. It changes the focus a lot with very little turning. Okay, this is the fine focus knob. It changes the focus very little with lots of turning. Right? So you'll only really use the coarse focus knob on the first lens, the scanning power lens. After that, it moves so fast that you'll go right past focus and you won't even know it. Okay, so you really only use it on the scanning power lens, which is this little one here, okay, which is what you use to scan the slide and find what it is you're looking for. Okay, that's really the only one we'll use the coarse focus knob with. When you go to the low power lens, that's the middle one, okay, you'll probably be using maybe a little bit of the coarse, but mostly the fine. And when you get to the high power lens, only use the fine focus knob. Okay? Again, the reason being, you'll go right past focus if you're using the coarse one, you won't even know it. And secondly, you can actually drive the lens into the slide with the coarse focus knob. Okay? It's got a lot of torque, okay? And it can break the slide and the lens. So we don't want to do that. Okay? We want to use just the fine focus knob, because look, like if you watch here, when I move this, I don't have to turn it very much for the slide, for the stage to move a lot. In fact, you can't even get one full turn of that knob, okay, before it's at the top or at the bottom of its range of motion. This thing, you can just keep turning it and turning it and turning it. It's like 100 turns, okay, to get it all the way from top to bottom. And it also will bypass the brake. The coarse focus knob has a brake to prevent it from going too far. Okay? The fine doesn't, because it doesn't have enough torque to hurt anything.
What's how much magnification is each lens?
I was just getting to that. So our first lens, the scanning power lens, magnifies 40 times. Okay? Our low power lens, the yellow one, the middle one, okay, magnifies 100 times, and our high power lens magnifies 400 times. Okay? So we get pretty good magnification. These are pretty good microscopes. Okay? You can see quite a bit with these, especially if you're experienced and you can, you know what you're doing. Right? Obviously, maybe not everybody in this class has actually used a microscope before. You probably don't have a lot of experience with one if you have, and that's okay. The first lab is mostly just about getting experience using the microscope, and hang how to use it, get things focused, find them, that kind of stuff. All right, final feature of the microscope. The nose piece or the lens piece rotates, okay? The eyepiece, rotates. And that's important for your group because what we don't want to do, what we don't want to do is be picking up the microscope and setting it down. Okay? Because I see people do this. They spend like 5 minutes and they finally got the thing they're looking at nicely focused. They feel good about themselves. And they're like, okay, you look at it. They pick it up, hand it to their partner, and set it back down. As soon as you do that, it's out of focus. Okay? That vibration knocks it out of focus. Right? And then they're like, well, you're no good. I can't see it. And then there's fighting. And it's no good. Okay? So what you want to do is one of you stands like this, and you're looking, you're focusing, and then just turn the eyepiece to your partner. Okay? Or get out of the way and let your partner look. Whichever. Okay? But this kind of eliminates the need to move around very much. Okay? Just make sure you use that. The more you pick up and set down the microscope, the more you're going to knock it out of focus. All right. So carrying techniques. Always carry the microscope with one hand on the arm, one hand on the base, as I demonstrated there. Okay? You always return the microscope with the scanning power lens in viewing position. That's just a courtesy thing. The next person expects that to be there. That's the one they're going to want to use. Okay? When using a microscope, always start with that lowest power. Okay? And here's the reason for that. When I'm looking through the microscope, okay, when I'm looking through the scanning power lens, my magnification is low, but my field of view is high. Okay? So imagine that when I look through the microscope on my scanning power lens, I see a big circle. Okay? When I move up to the low power lens, I increase the magnification, but I decrease the area that I can see because I'm magnifying what was there. So this is the scanning power, then this would be the area I see on low power. It'll be bigger, like it'll look the same, like the circle will be the same size, but I'll be looking at a smaller area, more greatly magnified. You follow me there? Okay. And then when I go up to the high power lens, I'll be looking at an area that big. It'll still look just as big, but it'll be a much smaller area, more greatly magnified. Okay. So I want to start on my scanning power because I can see more. It'll be easier to find what I'm looking for when I can see this much area than if I'm looking through this much area and trying to find what I'm looking for. Okay? My chances of finding it are pretty slim. It would be like doing this. You go to a Flames game, and your buddy texts you and says, yeah, I'm at the Flames game too. And you're like, well, where are you? He's like, well, I don't know. I'm in section whatever. And if you're like me, you don't know what the sections are. You know where to look. I'm wearing a bright yellow jacket. Okay, so do I like grab a high-powered telescope and start scanning the crowd one person at a time until I find a bright yellow hoodie? No. Okay, I look around with just the naked eye and look for anything yellow. When I see something yellow, then I go, is that him? And then I grab maybe my binoculars, okay, and I can see him. Okay, well, you laugh. Some people, they take binoculars to the game if they don't have good seats. Like me, because I never have good seats because I'm poor. Okay? If I get games, I'm in the sport check section, right? The cheap seats. Right next to the roof. That's where I sit. Okay? So then I would, check at a greater magnification. But when I look through those binoculars, I don't see the whole stadium anymore. I only see the area I'm magnifying, which is much smaller. Okay? That's what's happening here. So we always want to start with the scanning power to find what we're looking for. Now, once I find what I'm looking for, before I move up, I need to move the stage to make sure that object is centered. If I don't do that, okay, so let's say I was on scanning power and I found what I was looking for. It's that thing right there, whatever that is. Okay? Well, if I just move up to the next power, am I going to see it anymore? No, it's outside of my field of view. So I need to put it toward the center. There's a little thing called a pointer inside the microscope okay, that can help you with that. And it looks like a big black pointer, oddly enough, okay? That's what it'll look like through the microscope. It's actually a piece of brass wire, but through the microscope it just looks black, okay? And it points right to the middle. So it's supposed to be there. And I only say that because I've had like groups in the past where they keep bringing the microscopes. There's something wrong with this one. There's something wrong with this one. And I went through and like, I can't figure out what's wrong with it. What's wrong with it? Well, there's a big black thing in the middle. Pointer? Okay, so it's supposed to be there. That way nobody gets embarrassed now. Okay, because it was a bit embarrassing when that happened before. Like the pointer, and everyone's like laughing. And it's like, oh, I feel bad. Okay, so now that's supposed to be there. You all know. Okay, so get it on the end of the pointer, and then move up to the next magnification. All right, I am never going to ask you to label the parts of the microscope. Okay, there's never going to be a quiz or a question on a test or label the microscope. I don't care. Okay, you can use the microscope. Good. Okay? Now, at the same time, if I say, well, use the coarse focus knob, I need you to go here and not anything like that. You gotta at least know the parts somewhat so that you can use it and understand what I'm saying to you. Okay, need you to copy this into your notes because this is going to be important for us when we do our lab diagrams and estimate the size of the objects we're looking at through the microscope. So what this is showing you is the field of view for each lens. Okay? So when I look through that first lens, the scanning power lens, which has the red band on it, the distance across that I can see is 4,000 micrometers. Now this symbol here is mu. It's the Greek letter M. Okay? It stands for micro. Okay, so there are 1,000 micrometers in a millimeter. Okay, so this distance that I can see, okay, across the scanning power lens is 4 millimeters. So very, very small. Okay, everybody with me there? So less than half a centimeter. Okay. So if I was looking at something through the scanning power lens, and I know that this distance from side to side is 4,000 micrometers, and what I was looking at was that big, about how big is that? Yeah, about a millimeter, or about 1,000 micrometers. Okay? That's how we estimate the size of an object through a microscope. As long as we know what this distance is, we can estimate the size of objects. It's not a calculation, it's just an estimation. Now, for the low power lens, because for some reason it's disappeared, okay, the diameter for the low power lens is 1,600 micrometers, so 1.6 millimeters. And for the high power lens, the distance from one side to the other is 400 micrometers. Okay, or less than half a millimeter. Now, if you're trying to type this symbol on your computer or your phone or anything like that, there's a couple of ways that are acceptable. You can actually go to Insert, Symbol, and find mu, okay, and insert it that way. Or I'll also accept U, okay, the letter U. So if you want to put UM, okay, I'll accept that as mu M, because the symbol in Google like Docs and in Word doesn't look a whole lot different than a U, okay? So you can just put UM. Just don't put MM. Because if you tell me that something that's supposed to be a microscopic organism is 4,000 millimeters, you're telling me that microscopic organism is 4 meters. Okay? 4,000 millimeters is a lot different than 4,000 micrometers.
Can you just write like micrometers?
You could also type micrometers, yes. But just putting a U is really easy, right? That's why I just tell people just put a U. Okay, everybody all right with how we estimate the size of an object? We probably won't finish this next part today, but just so you get the idea, okay, there's a certain format for your lab diagrams. Fortunately for you, technology has advanced a lot in the last 25 years. You will not be required to hand draw your lab diagrams, because they're now called lab diagrams and not lab drawings, which is what they were called when I was in university. And I didn't do very well because I can't draw. I actually had to draw everything. It was terrible. It looked like a 2-year-old had drawn it. Okay? So now, what we do instead is, once you've got the object focused, take your phone, put your phone up against the lens, and it's going to take a little finagling, but you'll be able to get a good picture of it with your phone. And then you'll just insert the picture and then draw labels off of it, which I'll show you how to do in class tomorrow. Okay? How to make a lab drawing, lab diagram, with Google Draw, which is like the worst graphics program ever, but it does a good enough job for what we need, because mostly you're just going to have your picture that you took with your camera. Okay? And then, like I say, we're just adding labels and stuff like that to it. So, this is what we were looking at here just a minute ago. The scanning power lens is 4,000 micrometers. I just used a U. Okay, 1,600 micrometers, 400 micrometers. Okay, and we just went over how to estimate the size of the object. Okay, so what I'd like you guys to do real quick here is just give me quick answers to those questions on paper. See parts of an atom? I don't even know. A lot. No. Like, we're talking probably millions of times. But I would say a lot. They actually have some electron microscopes that they claim have resolved an atom, but they don't look like they don't look like the model. They're just kind of, that's kind of where we're at. That's probably what it is.